Hello, everyone. I'm Yan Benshen. I'm a first-year PhD student in Dr. Artie Singh's lab working on VOP breeding and high super phenotyping. It's my honor to introduce our first speaker in the afternoon, Dr. Malia Gehan. She's associate member and principal investigator in the Danforth Center. She's a very active member of the North American Plant Phenotyping Network, from where she has been awarded early career scientist. Dr. Gehan has been involved in the development of PlantCV software. Her research currently focuses on improving abiotic stress and answering questions as how plants perceive the temperatures, developing tools and pipelines of the high super phenotyping, and improve the measurement of plant phenotypes. She's also working on Brassica ACA for Bioenergy Corps and Heat Tolerant Corp for sustainable agriculture. Today, she will present what phenotypes matter, open challenges in plant phenotypes with PlantCV. Please enjoy me welcome Dr. Malia Gehan.
Thank you, Yan Ben, for that introduction. Are we going to get feedback? I'm going to put that down. I'll try not to stand directly behind the podium because I want you to be able to see me. Thank you, Yan Ben, for that really nice introduction. And thank you to the student organizers and to Corteva for the invitation to this meeting. I will preface this with saying I am definitely not a plant breeder. And so please do not expect that from my talk. And so my lab is generally interested in crop resilience to temperature stress, and it's really fortunate that I'm following Charlie's talk because I don't have to convince you that low temperature stress is an important stress to study. We also work on high temperature stress because of climate change, but we're generally interested in the environmental factors that limit the number of geographical locations that a crop can grow. We're also interested in the limitations to crops' growing season. So just as an example, if you were to be able to shift maize earlier, make it more cold or freezing tolerant, you might miss that heat or drought tolerance later in the season where it's most detrimental to those reproductive stages. So the way that we approach this question is really to study and utilize natural variation. And one challenging thing, I think this particular audience can really appreciate, is that if you want to make associations between genotype and phenotype, it often requires large-scale phenotyping. You can't get away from it. Be it manual or high throughput, it doesn't matter, you can't get away from it. And on top of that, there's a very tight relationship between abiotic stress and time, which makes all of your experiments exponentially large. So if you give a plant a low temperature treatment in the morning, you will get a different transcriptional response than if you gave that same cold treatment in the evening. If you gave a plant stress when that plant is a juvenile, you can get a different phenotypic response than if it was a mature plant. The duration of the stress matters. So an acute stress is going to be very different than a long progressive stress. And actually the time in which you phenotype those plants matter. So you might apply a plant stress and look hours later, not see any visible phenotype, but that might end up seeing a phenotype weeks or months later, depending on the crop you're working on. And so for that and many other reasons, I'm very interested in methods of high throughput phenotyping. And so I was lucky that at the Danforth Center, they've invested a lot in these types of technologies as a sort of nonprofit research institute. So we have one of these like fancy schmancy systems where you have a growth controlled environment growth chamber that can house 1,140 plants. Those then can grow in this system. and then get imaged every day. And for us, what's really nice too is that if you're interested in something like drought, you can do controlled watering treatments because those pots are getting weighed several times a day. There's also a second spout, so if you want to do a nutrient treatment, you can do that as well. So this system is great. It costs a lot of money. And even as somebody at the Danforth Center, it still costs me a lot of money to do these experiments. And so I'm also interested in low-cost methods of collecting similar types of data. And so we do a lot of Raspberry Pi imaging in my lab. So Raspberry Pis are these $35 computers that are the size of a credit card. And I think many of you are familiar with them here because Pat Schnabel's lab is obviously using a lot of those in the field. And you can get these $25 camera attachments. And what's nice is that they're cheap enough that we can throw them in these growth chambers. They can get humid and damp. And if they break, we can kind of say, okay, we'll replace them. They're cheap enough to do that. So we have over 200 of these in our growth chambers so we can test in different environments and look at plant growth over time at high temporal resolution. So we're taking images every hour in this case. We're also interested in different imaging modalities. So, this system collects RGB and near-infrared data. Our Raspberry Pi cheap and cheerful systems collect RGB data. But we're also interested in other imaging modalities like hyperspectral and thermal imaging, just as two examples. So this is a hyperspectral phenotyping robot prototype that we built in collaboration with Lematech when I started my lab. So it's hard to tell the scale of that thing, but it actually has a 7-foot reach. It's the size of the type of robots they use in the automotive industry to build cars. And we did that because the camera on this system is the most expensive part. It's not the robot arm. And so we wanted to be able to use one camera regardless of whatever plant architecture we had. So that's just kind of a summary of a very condensed summary of some of the systems we have at the Danforth Center. And so we're collecting all of this data. It's not on the same plants. We're a research institute with 31 different labs. And that means that we're looking at a variety of different crops and plant types and model systems. And we're also looking at a number of different imaging modalities. And so when we started this research back in 2013, and we looked at what commercial software was available to study image data at high throughput, there weren't too many options, especially not options that were, say, open source, that met our needs. And so there was commercial software that we had available to us that was made by software engineers, and we love software engineers. But they don't necessarily have the domain knowledge that plant scientists have to maybe define traits. And so just as an example, we had software engineers that defined plant height as this orange line. And there's plant material up here at the top and plant material down here at the bottom. But that's not what a plant biologist might consider the height of that plant. And so we needed flexible research tools that we could define so that it would make it easier for us to actually publish those results in the end. And so that's when, as postdocs, we started to develop a project called PlantCV. And so the idea behind PlantCV is that they would be phenotyping tools that would be modular and reusable so that they could be combined and recombined with ease, so you're not continuously reinventing the wheel every time you have a new imaging problem. It needs to be able to quickly extract biologically relevant data from images and sensors. And the tools, this is a key part, the tools needed to be usable by both data scientists, but also biologists. And that was because not every lab has the opportunity to collaborate with computer scientists. And so it needs to be usable by, say, the people who are collecting that data as well. And so at its core, PlantCV is a library of modular functions that you form into pipelines or workflows. And those workflows can be very complicated if you have complicated images, and they can be very simple if you have very clean images. And then within PlantCV, you have a way of parallelizing those workflows over large data sets that we assume are collected in similar ways. And then what you get out is a database of measurements associated with whatever metadata you have. We don't develop PlantCV in a vacuum. Wherever possible, we don't want to reinvent the wheel, so we are interoperable with other open source image analysis software packages. So PlantCV, again, it started in 2013. I think we actually had our first push to GitHub in 2014, so we're celebrating 9 years of PlantCV this year. And something that has been really important in making this a sustainable software project is that we spend a lot of time on the stuff that might not be super glamorous, and that is infrastructure. And that means that we can have continuous integration and continuous delivery of our software. So if I made a function today, you would have access to that function if I push those things to PlantCV today. And so we want to develop openly and give the community access to those tools as soon as possible. And what makes that possible, again, is the infrastructure that we've put in place. And so we're available via GitHub. Something that's quite unique is that we have a high level of code testing. What that means is that whenever someone builds a function, that code is tested. We make tests for that code to make sure that it's functional. And then if somebody makes a new function, say Yan Ben decided he wanted to make a new PlantCV function, if he pushed that function and then it broke something that I made, we can't merge those things until those conflicts are resolved. And so what this allows us to do, what this infrastructure allows us to do, is it allows us to have contributions from the community. So not just our core development team, but the community at large. So in that way, we can be more sustainable because we're not depending on one postdoc, staying in the lab to continue that software project on. We also spend a lot of time on documentation. And so we have documentation for every function in Read the Docs, and we also have interactive documentation. So you can try every function of PlantCV without even having to download the software. So I'm not going to read this list, but basically what PlantCV has is sort of all of the functionality you need to sort of segment and then extract image data measurements. We have some more specialized measurements, say graph-based segmentation, if you want to say skeletonize a plant and then measure things like leaf angle. And then some relatively simple but very powerful machine learning modules that help to classify and categorize images. So the funding agencies often want to know how many people are being impacted or how many people actually use your software because you're sending it out into the ether, out into the public. And so that's hard to necessarily describe, but we know that we have about 4,000 downloads per month via PyPy. That can be people just updating their PlantCV. But what's really important to us is that there have been 7 papers specifically on the development of PlantCV, but then 62 papers since we first pushed PlantCV that not just cite it, but actually use it in their methods. So these are people who are actually using PlantCV for their research. And this video is not playing right now. Maybe I could play it. But we're integrated with Jupyter notebooks so that you can get sort of real-time feedback if you're developing a workflow to see what that image analysis actually looks like. So here's just a few examples of what not necessarily us, but people out in the community are using PlantCV for. So people are using PlantCV as sort of an in-between step to pre-process and develop training data for, say, deep learning. So it can be a lot of effort to actually create these annotated data that you need for deep learning. But what's nice is that you could use PlantCV as to create those data sets so that you can actually use those deep learning methods that are so powerful. We're asked sometimes if PlantCV is useful out in the field, and right now we don't specifically develop tools for PlantCV out in the field, but sometimes out in the wild we see tweets of people using PlantCV on their field data. And that is going to be a focus going forward for PlantCV. We're developing more tools that are sort of interoperable with existing sort of field tools. Sometimes PlantCV is used for things that we do not expect. So these are some non-plant use cases for PlantCV. So someone used it to actually measure the cracks in sidewalks. So this was a material science paper. We never expected this application. And then somebody else was actually measuring vesicles in eyes. So it is not plant specific. We have a lot of documentation, and I think that is really, your software is only as good as the documentation that you have to be able to use it. And I think the fact that non-plant people are starting to use it shows that that documentation is useful. So we have a recently funded USDA grant to continue development of PlantCV, but also a big focus of this is to make it interoperable with other open source plant phenotyping software packages. So we're working very closely with Alina Zar's lab at the University of Florida because she has really nice tools for hyperspectral imaging. And so we've made a lot of the PlantCV segmentation tools interoperable with those tools, so you now can use those packages together. So if you have software that you are developing or functionality that you think would be useful in PlantCV, please reach out to us and we'd love to collaborate. So from here, I want to give 3 sort of separate use cases of how we are using PlantCV in my own group. The first use case is a focus on bioenergy. And so Brachypodium is this C3 model grass that is a model for bioenergy grasses. And so this is a project that is headed up by Ella Ludwig. She's a post-baccalaureate researcher in the group. So she did her undergraduate thesis with us and has sort of stayed on to continue her work before she heads off to graduate school. So in this project, Ella took 140 different accessions of Brachypodium that are mainly collected from Spain and Turkey. And then we did one of these very nice and fancy phenotyping experiments in our bellwether phenotyping facility. And we did 3 different stress treatments for conditions total. We had a control treatment, a drought treatment, a heat treatment, and then the combined stress treatment. And I don't have time to go over all of the trait data that we extracted from this via the images, but I'm going to focus on one trait in particular. And that is what we're calling a tissue damage or a stay green trait. So here we are, if you can see the pictures on the left side, we're using the PlantCV module. It's a naive Bayes machine learning module, and you can basically take your image, segment your plant, and then use that naive Bayes classifier to classify each of those plant pixels into whatever classes you want. In our case, we're classifying it in between damaged versus undamaged tissue. You could also use the same tool for if you had a biotic stress, or other people have used it for abiotic stresses as well. So again, we're classifying those into those 2 different categories. And what I'm showing here, this is a summary of all of that analysis. So 200,000 images in one graph. This first panel is control, the second panel is drought, third panel is heat, and the last panel is heat and drought. And the y-axis is all of those 140 accessions, and the x-axis, again, because it's non-destructive, is time. So we're looking at the change in the tissue damage over time for all these plants. And what we saw, this was a result that I like to show because it surprised us. We naively thought that if you give a plant 2 different stress treatments, heat and drought, you're going to be more stressed than if you had the heat stress or the drought stress alone. Very obviously, you can see that there's way more orange in this graph, indicating that we have a lot higher level of tissue damage, specifically in our heat-treated samples. So these are heat-treated but well-watered. And so our hypothesis is that maybe these samples are undergoing both heat and drought. Maybe heat is priming those plants to expect a drought treatment or a drought condition, and that actually when you have a heat with well-watered, maybe you're more susceptible to an osmotic stress. It's one hypothesis. So Ella then wanted to know, we have all of these accessions and they all have a climate of origin. So are the different traits that we're looking at, are those correlated, or how well those plants do, is that correlated with the climate of origin of those plants? And so Ella spent a lot of time fighting with WorldClim data. I'm not showing the full analysis where she looks at the whole population of all of the climates of origin. But what I want you to get out of this is that what we saw was that there was actually strong correlation between climate of origin and a lot of the traits that we were interested in. So here I'm showing percent damage. We also looked at things like height and total area of the plant, which is an estimate for biomass. And so here on the top, we have a line that we've picked out, BDTR10E, and on the bottom is a line BDTR5J. They're both Turkish lines. So this, we always like to look at the images as a gut check to make sure that what we've pulled out, via, just looking at large tables of data, represents what we see in the plants themselves. And so here's a line that's relatively robust across all of those stress treatments. So under drought, heat, and drought, you can see it does relatively well in comparison to this line 5J, where you can see that specifically in height, this plant is basically dead. And whereas in the heat and drought conditions, that plant is small, but it's still green and photosynthesizing. And so we look at these two lines in terms of their climate of origin. This line comes from a warmer area. And so it's potentially why, it's doing better. It's potentially locally adapted for higher temperatures in comparison to this bottom line that comes from cooler climates. So again, the idea is we take all of this trait data and then we can look over time doing genome-wide association mapping. This is an endpoint. And so this is doing GWAS for percent damage. And I want to specifically just point out this particular hit that we see. And so that one is a hit for percent damage under heat. And it is a gene that's also happens to be known to be affected under temperature stress. So this was the closest gene to that SNP. We didn't look at the many genes that are obviously surrounding that, which also could potentially be putative candidates. But that is the closest one, and it happens to be sort of an interesting one to maybe follow up on. So Ella, as part of her work, is following up on all of our sort of gene candidates. So making transgenic lines to actually test some of these genes of interest. So now I'm going to switch to our next story. So this is a relatively new project that we started in 2021. And this is on another bioenergy project. It's on high oil tobacco. So instead of thinking of like an oilseed crop like canola, where you have to wait for the full life cycle of the plant until you get that plant product, the seed, this is engineered tobacco that actually produces oil in its leaves. So it's 30% of leaf weight is actually oil. And we did not make these lines. These are lines that were made by CSIRO in Australia. And they're really, really cool. And Kevin Chu and Doug Allen's group did cross-sections of these leaves, and you can see in green, that's actually lipid. So in the wild type plants, red is that chlorophyll, blue is your cell walls, and then green is lipid. So we want to compare your wild type cross-section in leaves, your high oil. So there's a lot of oil that's being packed into those leaves. So one interesting potential application for this is, well, why don't we have our tobacco growers down in North Carolina, instead of growing tobacco for cigarettes, maybe they can be growing tobacco as a bioenergy product instead. So our part of this project, as people who are interested in abiotic stress, was really to understand what happens to the plant product, in this case oil, under different abiotic stresses. So we were looking at low temperature stress, but Katie's project in particular was looking at high temperature stress, especially since we're interested in growing this, say, in North Carolina. So the first step to this project was, Katie did, was just to do estimations of dynamic estimations of whole plant leaf transpiration using thermal imaging. And so she did this under high temperature conditions. And we previously didn't have tools for thermal imaging in PlantCV, and so we needed them. And so we developed selfishly. And so we developed tools for thermal imaging that are now sort of available in PlantCV. So this is the results for that experiment. And in the red line, you can see what the chamber temperature was set at. And then the blue line is wild type leaf temperature, and then the green is high oil tobacco leaf temperature. So we did that under controlled temperature conditions, but then also a very high temperature heat stress condition. And what you can see consistently under both the control and the high temperature experiment is that the high oil tobacco has a hotter leaf temperature. So suggesting that potentially we might have some sort of stomatal phenotype maybe. And so because of this data, we started to look more closely at the surface of those leaves. And so Katie did impressions with this dental putty on the surface of those leaves. It's a non-destructive measurement. And then you, on the dental putty impressions, you brush nail polish, and then you can get images that look like this once you image them. And so at first, we were like, okay, how are we going to quantify the stomatal aperture in these images? And you could use a tool like ImageJ, and that's what we started doing. But it's very tedious. You have to sit there and like drag the line across the stomata and see how wide they are. And so it takes a lot of time. And so again, here is where we take advantage of the fact that we are also interested in high throughput phenotyping and that we develop tools. And so this is a new interactive point and click tool in PlantCV. And so you don't have to drag anything. You just blop, 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 blop over all of those stomata. And you don't have to be very exact. And what you get out is something that looks like this. So you get measurements of each of those stomata. And eventually, when we have enough images, then we can do deep learning. And so that hopefully we don't have to do even the clicking. So we get, again, we're using all of our toolkits available to us. We didn't have this tool available, and now we made it. So we can get size and shape and morphology of that stomata. And just as an aside, we try and create tools that are non-specific. And so this tool, we needed it because we wanted to measure stomata, but we wanted to also try to use the same tool for other applications. And so we have an interest in quinoa in the lab, and quinoa is particularly salt tolerant. And actually will have, it has organs, modified trichomes on the surface of those leaves that will actually excrete salt so that they look like balloon-like structures. So we wanted to actually be able to quantify the number of salt bladders on a salt-sensitive line in comparison to a salt-tolerant line. And so we used that same tool. We made very minor modifications to that tool. And so we can actually quantify the number of salt bladders using that same stomatal tool here. So same tool, different application. And so here we can see there's more salt bladders on the surface of that salt tolerant line. Okay, we're going to go back to the tobacco data. So after quantifying the stomata using our stomata tool, we find that not only are there more stomata in the wild type, they're wider, whereas the high oil tobacco has less wide stomata, yeah, they have less stomata and they're also narrower. So why is that? So we wanted to know, okay, that's great, that makes sense. It follows the idea that these plants are able to transpire more. They have wider stomata, so that's why we had lower leaf temperature. But why are these stomata less open? And so again, you remember that first slide that I showed with the cross-section of the tobacco. This is now looking at the surface of the tobacco leaves. So here you can see in the wild type, there's a lot of oil, which is in green again, packed into the trichomes of that surface of those leaves. And then you'll notice that there's a very thin line here that's sort of the guard cells in the stomata. But here in the high oil tobacco, you see a lot more oil packed into the stomata. And so we have a couple of hypotheses then that perhaps there's actually a physical barrier for those stomata to close because they're packed with these oil bodies. So stomata are often also very important to photosynthesis. So we wanted to actually measure carbon assimilation and other photosynthetic traits in these plants. So we went from thermal imaging high throughput to something a lot slower, which is taking live core measurements on these plants. So it's really nice to be able to do live core measurements because they're so detailed and we get a lot of information, but it's obviously not a high throughput method. And so Courtney did these measurements in collaboration with Katie. She was an undergraduate researcher in the lab. And what we can see is that these high oil tobacco lines have significantly lower carbon assimilation rates in comparison to wild type. Not only that, but if you look at the slope of these lines, when you change to highlight, you see that the stomata are less responsive in comparison to the wild type lines. So we have two hypotheses here as to why, again, we have this difference in stomata opening and closing. The first is that they might not be closing because ATP production is tied up because there's so many oil bodies tied up as tag. Or again, this mechanical issue, Katie likes to describe it as if you had an inner tube filled with basketballs. So you can't squeeze that inner tube shut because you have all of these oil bodies in that thing. So you can imagine that having a stomata phenotype, you might have a significant impact on the stress tolerance of these plants. And so this is very preliminary data. Where not even labeled, you can see that, so this first one's wild type, the second one is high oil, wild type, high oil, wild type, high oil. And what we're going to do is we're simply going to just stop watering these plants. And then we're going to look at these over several days. And so if this video will play, you see that there's the circadian oscillations of the leaves. The control plants sort of droop right away, but they kind of come up the next day. And then the high oil lines, they droop, so they're likely losing water off of their stomata that are more narrow, but they cannot recover as well as the wild type lines. So again, the stomata phenotype is that we got after doing a high temperature experiment also has this drought phenotype as well. So we're following up on this and some other heat stress measurements looking at the lipid content under high temperature as well. Okay, and so in my last few minutes, I'm going to tell a very incomplete story. on a project we have that's on an NSF Rules of Life project. And I wanted to tell the story specifically because it connects to the brachypodium story and the high oil story that I just told. And so we have this project that we're interested to see what happens under high CO2 conditions because of climate change and rising CO2 conditions. So we're growing many different species of plants. So here I'm showing Arabidopsis under either ambient CO2 conditions or high CO2 conditions. Then we are taking the progeny of those plants, and we're seeing how if these phenotypes from generation 1 persist to generation 2. So if the high, just an example, if the high CO2 lines were larger in generation 1, does that phenotype persist into generation 2, even though they are in ambient CO2 conditions? And so we're not just doing this in Arabidopsis, although there's a lot of advantages of doing that. We are doing this across several different species of plants. So we're doing this in Arabidopsis, including many mutants, to try and tease apart mechanism. We're looking at this in a C4 model grass Soteria. We're looking at the tobacco line, including that high oil line that has a stomatal phenotype. So we're looking at that under high CO2. We're looking at brachypodium, and we're specifically choosing Ella's favorite lines that were robust under those different stress treatments. We're looking at moss, and we're also looking at rice. And so one challenge with this particular project is that we are collecting all of this data using this Raspberry Pi phenotyping system. And so we're in growth chambers, and eventually these plants grow over each other. And for a computer, that is difficult. A computer doesn't know that these are supposed to be individual plants and not one large plant. And so many people have approached this problem, and they have done the work to either make synthetic data or to trace these images, right, so that you can actually segment them. But that's a lot of effort, and we have to do a lot of different plant species. So we don't have the time to make these data sets. So again, Jorge developed this PlantCV module that applies a watershed algorithm spatially and across time so that we can propagate labels from when the plants aren't touching to when they are touching. And so with that, you can see in this last image, without doing any training, we can actually segment these plants and get more data out of that data set without having to do deep learning. And so we wanted this tool to be, again, to be able to be used not just on one species, but again, we can use this on Soteria in this case that has a very different architecture type. So just as an example of, so we recently published a paper out of this work, and by recently I mean in the past week, that this paper is now out at New Phytologist. And so again, we were growing this, these are Arabidopsis data only. So we were growing under ambient CO2, and then not surprisingly, the high CO2 plants were larger. But was interesting that phenotype persisted to the second generation, even though they were under ambient CO2 conditions. And so we used a bunch of different Arabidopsis mutants to actually determine which RNA-dependent DNA methylation factors were required for this phenotype. So where is this project going next? This project is, we're interested in getting more data out of just even the Arabidopsis data, but also we're analyzing all of the other species of plants that we're looking at. And so again, one challenge here, you know, when we were looking at Arabidopsis before, we're just treating it as a bunch of pixels. And we want to be able to actually say track the development of individual leaves over time. And so people have worked on this problem, and you know, we were not the first ones by any means to solve this by using masked RCNN. And so you can actually do a nice segmentation of these individual leaves. But one thing you'll notice is that the color of those leaves is jumping all around. And that's because leaf 1 from image 1 is not linked to leaf 2 from leaf 1 from image 2. And so we wanted a simple way to, within PlantCV, to link those leaves through time. And so Danny developed that. And so we have a new time linking module in PlantCV that's sort of robust, at least for the species we've tested so far, so that we can actually link leaves through time. And so we can get leaf emergence and then also tracking individual leaf size. So that in that way, we can get more data out of the single data sets that we already have. Okay, so just in summary, we develop what I hope are usable, user-focused tools for open source plant phenotyping. We have used these tools to identify and utilize natural variation in Brachypodium to identify genes that are potentially important for abiotic stress. We use different imaging modalities to identify potential mechanisms for difference in high oil tobacco stress. And we were looking for patterns of phenotypic change across similar, different species in similar environments. So with that, I will thank the people who have done all the work. They're highlighted there in bold. And I will also say that we are hiring. So if you're interested in some of the work we're doing, we're looking for postdoctoral researchers or graduate students in the future if there's any undergraduates in the room. Yeah, I'm happy to take any questions if there are any.
Honored to have a chance to talk with you during my poster session. And since we both working in a similar direction, image-based HIGH-THROUGHPUT phenotyping, so I have 3 questions here. Please forgive me, I occupy more time. So the first question is, have you ever applied the PlantCV for other species for the, so you study the leaf stamina. Have you only apply for one species, here? Have you ever considered to apply it to other species, especially with a higher, taller canopy?
Are you talking about the thermal imaging, or are you talking about the stomato tool specifically?
The stomato specifically.
Oh, yeah. So it's a relatively new tool. So we ourselves haven't used it, but I would assume that if you're collecting the data in a similar way, so especially if you're doing dental putty and then the sort of nail polish method and doing that sort of same microscopy method, that you'll be able to use that same sort of point and click tool. Yeah, most of our tools are pretty species agnostic.
Yeah, so and then my second question will be related to the first one is, we know the importance of the leaf stemma for the photosynthesis, and you in your result you shows the leaf temperature will also be adjusted by the stemma. I was thinking if for the taller plants, they have more leaves, whether the shading will affect the temperature at bottom leaves. So the different leaves across the whole canopy have a different photosynthesis rate.
That's a good question. The question is, do the plants have different leaf number? So Katie actually spent the time to count the leaves of the plants, especially at the stage she was looking at, and at least the leaf number is the same, even though the plants, they are smaller, which is not surprising considering the differences in in carbon assimilation. Yeah.
Thank you. My last question is for the time linking module, and you can keep track of the leaf size changes, but that your pipeline can keep track of the order of emerged leaves. Yeah, that's
Yeah, so that, that particular tool assumes that you have a time series. And so because it is linking it, you do get leaf 1, leaf 2, leaf 3 emerging.
Yeah, but so you keep track of the leaf from the image, but the leaf position could change. So it's kind of hard to keep track of the.
Yeah, at least in the case that we are doing, we're imaging frequently enough that the amount of movement from image to image is not great. So we found that like you need a minimum number of images from image to image so that you get a good intersection over union of those two before and after. Does that make sense?
Thank you.
Yeah.
Thank you, Dr. Gehan, for your presentation. I have a question about you showed the short video tracking the development of leaves with like RGB sensor. I'm curious, can you do that with your hyperspectral sensor too?
It doesn't, the time tracking series doesn't take, there's no color information that it's being used. So as long as you had segmentation, then you shouldn't have, you don't need it. You could make a pseudo RGB from your hyperspectral and then go from there as well. But
it's probably too much data collection from.
You could do it. It would be intense depending on the size of your hyperspectral images. For sure.
Perfect, thanks.
Yeah.
Thank Dr. Gehan for your presentation. Yeah, I really love PlantCV, but as you mentioned, I do HIGH-THROUGHPUT phenotyping in the field, so I look forward to the next, especially I see that you are kind of having some integration with field image R, which I currently use.
Yeah, so just as an update to that, so Philippe, obviously he's moved on to bigger and better things. And so the PlantCV team is now helping on the support end of that. And so if new functions for field image R are needed, we'll likely develop those within sort of PlantCV framework just because that's what we're comfortable with in terms of new functionality. Yeah.
Yeah, well, that would be great. One new function we are looking forward to is the segmentation, especially for field where the plots are not well aligned. But I have one question, because you mentioned how you measure traits throughout development or throughout time. So you show one GWAS plot. Have you looked at the effect of these SNPs across time, how they're changing?
Yeah. So it's definitely not the same. So we've looked through time. We often see overlapping SNPs between the stresses, and those are at the same developmental time, rather than seeing the same SNP between time points. Yeah.
Okay, thank you.
Yeah.
For your presentation, and let's join me. Thanks again of Dr. Gehan.